Good afternoon. Throughout today's presentation, we will explore glycolysis and gluconeogenesis pathways, focusing on enzyme aldolase and its regulation mechanisms. Glucose metabolism is central to energy production in our bodies. Glycolysis and gluconeogenesis are two critical pathways that work together to maintain glucose levels, depending on the body's energy needs to maintain homeostasis. Throughout this presentation, we will overview glycolysis and gluconeogenesis and their reciprocal regulations. We'll also be touching on aldolase in both glycolysis and gluconeogenesis, the three isoforms of aldolase and their expressive pathways, as well as the regulation of specifically aldolase B expression and transcription factors, and the transcription regulation mechanisms. We'll then close with current clinical research that is relevant to these topics. Glycolysis is both a metabolic pathway and an anaerobic pathway for energy source that is involved in nearly every type of organisms. Although it needs no oxygen, hence the anaerobic aspect, it is the first step in cellular respiration. Glycolysis breaks glucose into pyruvate, yielding energy as ATP and NADH. This pathway occurs in the cytoplasm of cells and is vital for producing energy, especially in tissues like muscles and the brain. Referencing the image, glucose and 2 ATP molecules are consumed to allow glycolysis, producing 4 ATP and 2 NADH molecules, and 2 pyruvates for every 1 glucose molecule used. The pyruvate and versatile molecules which is a versatile molecule, can then be utilized in the citric acid cycle or serve as a precursor for other reactions, showcasing its importance in cellular metabolism. Often we consider glycolysis broken into 2 phases, the investment phase and the payoff phase. The investment phase is a critical part of glycolysis. It's like the initial stages of a project where the groundwork is laid and the foundation is set for the rest of the work. Glucose has 2 phosphate groups added, and the glycolysis starts with hexokinase phosphorylazing glucose into glucose 6-phosphate. This is the first step in transfer of phosphates, where the first ATP is consumed. Again, we talked about how 2 ATPs have to be consumed in order to start the process. This is that first one. This is also irreversible. When the phosphorylation occurs, it traps the glucose in the cell, not allowing it to pass the membrane. The G6P isomerizes into fructose 6-phosphate. This fructokinase is what adds the second phosphate. This uses the second ATP and phosphorylates the F6P into fructose 1,6-biphosphate. This is also irreversible. The fructose 1,6-biphosphate is broken down via aldolase, converting it into DHAP and G3P. The DHAP is turned into G3P via triacylphosphate isomerase, as they are within equilibrium, allowing each other to transform back and forth from one another. You will notice there are a lot of acronyms and a lot of words associated with numbers, so please bear with. Then we have the payoff phase. At the beginning of this phase, there are 2 3-carbon sugars per 1 glucose. The enzyme glyciderate aldehyde 3-phosphate dehydrogenase metabolizes the G3P into 1,3-diphosphogluconate. This is done by reducing an NADH positive into NADH. The 1,3-diphosphate glycerate loses a phosphate group via phosphorylate kinase, making 3 phosphoglycerates. This creates 1 ATP through substrate level phosphorylation. 2 ATP are produced, 1 from each of the 3 carbon molecules. The 3-phosphoglycerate turns into 2-phosphoglycerate by phosphoglycerate mutase and then enzyme analase converts 2 phosphoglycerates into phosphophenolphthaleate, which is considered a PEP. Finally, pyruvate kinase turns PEP into pyruvate and phosphorylates ADP into ATP through substrate level phosphorylation with the final product of 2 more ATP. This is also irreversible. The entire process yields the input of 1 glucose molecule is ATP, 2 ATP. The output is 4 ATP and 2 NADH and 2 pyruvate molecules. To ensure glycolysis continues, NADH cycles back to NAD+. The absence of NAD+ will halt the payoff phase, creating a backup of glycolysis. That was a lot of talking and a lot of information. I understand this image is very simple. Throughout the entire presentation, we will go back and touch back on every single one of those things that have been mentioned. So just bear with and kind of keep them in your back pocket for now. Understanding that glycolysis can undergo either anaerobic or aerobic state, it is vital to understand the differences in these processes. Under aerobic conditions, pyruvate plays a pivotal role as it enters the citric acid cycle and undergoes oxidative phosphorylation, resulting in the net production of 32 ATP molecules, very different from what we just discussed. In the anaerobic conditions, pyruvate is converted to lactic acid or lactate, which produces 2 ATP molecules. Here in the image, we note that glycolysis occurs in the cell's cystoles. In aerobic conditions, the pyruvate, which is taken from glucose, enters the mitochondria to undergo its oxidative phosphorylation. This is shown in the green or on the right-hand side of your screen. In the blue section of this image, or to the left, we see the anaerobic conditions, a result of pyruvate again taken from glucose, shown in the top box of the flow chart, staying in the cytoplasm of the cell to be converted to lactic acid with the byproduct of alcohol and carbon dioxide. This is done via the enzyme lactate dehydrogenase through the process of fermentation. Gluconeogenesis is the reverse of glycolysis. This is because pyruvate is converted back into glucose. The primary stimulus for gluconeogenesis is low blood sugar. Starting with pyruvate, the reaction involves in gluconeogenesis are where in the mitochondrion, pyruvate carboxylase converts pyruvate to oxaloacetate. Pyruvate carboxylate requires ATP and the coenzyme biotin for activation. This conversion is the first step that reverses the non-equilibrium reaction catalyzed by the glycolytic enzyme pyruvate kinase. Oxalic acid is converted to maltate before it crosses the mitochondrial membrane. Maltate is converted back to oxaloacetate once in the cytosol. In the cytosol, the PEPCK decarboxylates oxaloacetate, which is then rearranged from phosphophenols or that PEP. The PEPCK requires GTP and magnesium ions for activation. This transformation is the second step that reverses the non-equilibrium reaction catalyzed by pyruvate kinase. Again, we are going to see these terms show up in another slide, but just follow along with the image that is on this slide as well. The analase hydrates PEP to form 2 phosphoglycerates. The phosphoglycerates mutase converts 2 phosphoglycerates to 3 phosphoglycerates. Phosphorylase kinase phosphorylates 3 phosphorylate to form 1,3-biphosphorylate glycerates. This reaction also requires ATP. Following along, we have the glyceraldehyde 3-phosphate dehydrogenase reduces the 1,3-biphosphoryl glycerate to glyceraldehyde 3-phosphate. The reduced nitroamide adenine dinutrate, or NADH, is the electron donor. Triose phosphate isomerizes the glyceraldehyde 3-phosphate to form the DHAP. Aldolase combines glyceraldehyde 3-phosphate and DHAP to form our fructose 1,6-phosphorylate. All of these will be talked about again. Fructose 1 and 6 biphosphate dephosphorylates fructose 1,6-biphosphorate to form fructose 6-phosphate. This step reverses the non-equal reaction catalyzed by the glycemic enzyme phosphofructase 1. Then we have the phosphohexo isomer converting the fructose 6-phosphate to glucose 6-phosphate, and that dephosphorylates glucose 6-phosphate to form the glucose, which can enter the bloodstream freely. So now we have gone back to glucose. This last reaction requires the non-equilibrium reaction catalyst by the gluconate glycosidic enzyme hexanase. This process is critical, especially during fasting, as it allows the body to produce glucose from non-carbohydrate sources, maintaining a blood sugar level. This metabolic reaction occurs in the cytosol and the mitochondria of the cell to maintain a homeostatic blood sugar or blood glucose level. During a fast, the hepatic glycolysis is used to primary source of glucose. However, this only occurs in roughly the first hour of the fast. Glucose stored as glycogen gives the body the glucose needed for roughly the first day of fasting. However, after starvation sets in, the gluconeogenesis and the glycolysis contribute equally to the blood glucose supply. The amount supplied by the glycogen will eventually and then rapidly decrease, and the glucose from the glyconeogenesis will increase, keeping that total level constant, again, maintaining homeostasis. These two pathways are tightly regulated to prevent simultaneous occurrence, which would be insufficient. Essentially, while one process occurs, the other one is turned off. Bear with me as we look at this image yet again. Following glycolysis from the top center to the left, we can see that when there is a low amount of ATP, the cell wants to produce a large amount. Makes sense. The tiny purple and green circles on the top left of the flowchart represent the effectors that activate glycolysis. So here's the breakdown. Phosphofructokinase, which we've mentioned before, and pyruvate kinase, again mentioned before, are the heroes that step up to produce pyruvate, while PEP, carboxyl and pyruvate carboxyl are the ones who take a back seat. So while one is activated, the other one is essentially deactivated. On the flip side, fructose 1,6-biphosphates, PEP carboxylase, and pyruvate carboxylase are put on pause. That would be the ones that are now deactivated. Enzymes like PFK1 in glycolysis and FBPase1 in gluconeogenesis play opposing roles, helping control the metabolic flow based on the cell's energy status, whether they have high energy or low energy. Glycolysis is a carbohydrate process of glucose hydrolysis needed for energy and biosynthetic intermediates, whereas gluconeogenesis is a glucose production process essential for maintaining blood glucose levels during starvation. Two enzymes that regulate the irreversible steps of these processes are the pyruvate kinase, or the PK, and the phosphoenolpyruvate carboxylase, the PEPCK, both of which we've talked about multiple times. These catalyze the last and subsequently first step of glycolysis and gluconeogenesis. Aldolase is a key enzyme in glycolysis. It catalyzes the conversion of fructose 1,6-biphosphate into 3 carbon molecules, DHAP and G3P. This step is crucial for splitting that 6-carbon glucose molecule into 2 smaller molecules that can be metabolized. This occurs during that 4th step of glycolysis. Aldolase is also involved in other cellular functions, including muscle maintenance, cell shape and mobility, regulation, citration muscle contractions, and actin filament organization. And here we have an image of what that step 4 looks like. Interesting, aldolase also plays a role in gluconeogenesis, catalyzing the reverse reaction. This reversibility is essential for the flexibility of these metabolic pathways, allowing the cell to adapt to different energy demands. Aldolase is a cytoplasmic enzyme that catalyzes the reversible conversions of fructose 1,6-biphosphate into dihydroacetone phosphate, or DHAP, and the G3P. This reaction is a key step in the glyconeogenesis pathway, which synthesizes that fructose 1,6-biphosphate from those 3 triose phosphates. Aldolase also works on 6 other reversible reactions in glycolysis. Different isomers of aldolase exist, and each prefer either or both substrates depending on its role in gluconeogenesis versus glycolysis. For example, aldolase B is found in tissues with functional glyconeogenic pathways and is involved in both glycolysis and gluconeogenesis. Aldolase A and C are mainly involved in glycolysis, with aldolase A being expressed in muscles in the brain and aldolase C being expressed in the brain. So this image here is just a very big, complex image of everything that we've already talked about that really highlights all of those different components that have been discussed throughout the last few slides. And it shows how they transfer from one to another and how aldolase comes into play for them. The expression patterns of these isomers are tightly regulated during development and in response to the metabolic needs of different tissues. For example, aldolase B is highly expressed in the liver, which plays a key role in both glycolysis and gluconeogenesis. Aldolase A is expressed predominantly in muscle tissue and is also found in the brain and other tissues that have a high demand of energy. Its function is to play a critical role in glycolysis by catalyzing the reversible cleavage of fructose 1,6-biphosphate into the DHAP and the G3P. In muscle tissue, it supports rapid energy production during physical activity. Aldolase B expression is mostly in the liver and to a lesser extent in the kidney and small intestine. It functions in both glycolysis and gluconeogenesis. In the liver, it plays a role in fructose development and particularly in the cleavage of fructose 1-phosphate, and is essential in it's an essential step in converting dietary fructose into intermediates that can be used in energy production or storage of glycogen, which, as we discussed before, during a fast or during starvation is where we get our glucose energy from. Full circle. And then we have the aldolase C, which is mainly expressed in the brain and in the nervous tissue. Similar to aldolase A, aldolase C is involved in glycolysis, particularly in the nerves, where it supports the brain's high energy demand and also plays a role in maintaining the brain's metabolic functions. So in summary, aldolase A is high in muscles and in the brain and other tissue. B is in the liver and has a presence in the kidneys and small intestines, and C is in the brain and other nervous tissues. Each isomer's expression is tailored to meet the metabolic demands of the tissues in which they are predominantly found, ensuring efficient energy production and metabolic regulation in different physiological contexts. Several key transcription factors regulate the expression of aldolase B. These factors ensure that aldolase B is expressed appropriately in tissues like the liver, the kidney, and the small intestine, and that its expression responses to the body's metabolic and hormonal needs. Similar to those regulation factors we discussed on the previous slide, the primary transcription factors involved in the regulation of aldolase B are HNF1, and it is a liver-enriched transcription factor crucial for liver-specific gene expression, specifically those that obviously include aldolase B. Its function is to bind to specific sites on the promoter region of aldolase B gene and promote its transcription in liver cells. Then we have the HNF3, which is also known as the FOXA protein. And these transcription factors are involved in the liver development and the regulation of metabolic genes. They bind to the aldolase B promoter and collaborate with other factors to maintain basal expression levels of aldolase B, especially in response to metabolic change. We then have the C-EBP, which is an enhancer binding protein. And this is involved in regulating genes linked to metabolism, inflammation, and differentiation. And they function to enhance transcription of aldolase B by binding to its promoter region, especially during fasting when glyconeogenesis is activated. The D-site binding protein is a transcription factor influenced by the circadian clock and is involved in liver metabolism. It functions to regulate the circadian expression of aldolase B by binding to its promoter region, aligning aldolase B levels with the daily metabolic cycles. It also has a hormonal influence on transcription factors, such as insulin, where it suppresses the expression of aldolase B in the feed state, reducing the glyconeogenesis. And then again, in the glucagon and glucocorticoids, it increases the expression of aldolase B during fasting, promoting gluconeogenesis. These transcription factors work in concert to regulate the expression of aldolase B, ensuring that it's expressed in the right tissues at the right time in response to the body's metabolic and hormonal signals. The transcriptional regulation of aldolase B involves several mechanisms that ensure its expression is tightly controlled, particularly again in the liver, kidney, and small intestines. These mechanisms include the interaction of the transcription factors with the aldolase B promoter, modulation by hormones, and influence of circadian rhythms. Looking at these different promoter binding transcription factors, we have, again, they're all going to be the same as listed in that last slide, but the HNF1 binds to specific sites of the gene promoter, and it recruits other co-activators and transcription machinery that enhances the transcription of that aldolase B in the liver cells, and the outcome is ensuring that the liver-specific expression of aldolase B is correct. Then we have the HNF3. Again, that's that FOXA protein, and that binds to the aldolase B promoter region, acting as a pioneer factor. It opens up the chromatin structure, allowing for DNA to become more accessible to other transcription factors and transcriptional machinery. This outcome maintains the basal expression of aldolase B and primes the gene for activation in response to any metabolic changes. Then we have that C-EBP protein, again binding to that promoter on aldolase B, and these proteins act as transcriptional activators, especially during states of fasting, promoting that transcription of aldolase B with the outcome of upregulating the expression during gluconeogenesis. Then we have that D site binding protein, and this again promotes or is attached to the promoter and regulates the circadian clock. Binding influences that rhythmic expressions of the Aldolase B and aligning it to the levels within the daily metabolic systems. This ensures that we follow the circadian rhythm with peaks and troughs throughout the feeding and fasting cycles. And again, hormonal regulation is involved with insulin. Signaling leads to the activation of transcriptional receptors or inhibitions of transcriptional activators, binding to that promoter site. This reduces the transcription during feed state when the glucose levels are high and gluconeogenesis is not needed. The outcome of that is that it now downregulates Aldolase B expression in the response to that high glucose levels. And then we have the glucagon and glucocorticoids, and these activate transcription factors, and these hormones are increased during fasting, leading to the upregulation, promoting gluconeogenesis, and the upregulation of Aldolase B expression during fasting to maintain that homeostatic blood glucose level. Then we have the transcription factors like that FOXA protein, which is the pioneer factor, binding it and altering the chromatin structure. And this process makes DNA more accessible, influencing the expression levels of Aldolase B. And this facilitates the transcription of Aldolase B by making the promoter region more accessible. It also has histone modification like acetylation and methylation of the histones around the Aldolase B gene, and it can either enhance or repress the transcription. And this promotes transcription. It typically loosens chromatin structures, while the histone methylation can either activate or repress gene suppression, depending on the specific needs. The outcome being it modulates the transcriptional activity during response to cellular signals. And then lastly, we have the circadian clock. This gene regulates the expression of the transcription factors such as DPP, and influences that rhythmic expression and syncs it to the daily metabolic cycles, again, having the metabolic processes based on the time of day. Disruptions in aldolase function or regulation can lead to metabolic disorders. The aldolase isoforms play crucial roles in cellular metabolism, and their dysregulations can lead to various clinical conditions. Understanding the clinical significance of these isoforms A, B, or C, and the diseases that are associated with their dysregulation is essential for diagnosing and treating related disorders. Specifically, when we're looking at aldolase A, which is crucial for glycolysis, particularly in muscle tissue, and supports that rapid energy production. It's also essential in the brain and other tissues, to making it essential for an overall energy metabolism. Some of the diseases that are associated is myopathy, and this is a mutation or deficiency of that aldolase A and can lead to a glycocytic myopathies, which are characterized by muscle weakness, cramps, fatigue, especially during exercise. Patients may present with elevated serum aldolase levels indicating muscle damage. There's also the hemolytic anemia, which is in rare cases. There's a defect in that aldolase A, and it can lead to this anemia where the red blood cells are destroyed faster than they can be reproduced, meaning that there is a result in fatigue, jaundice, and other anemia-related symptoms. When looking at aldolase B, it is essential for that fructose metabolism in the liver, kidney, and small intestine, and plays a role in breaking down that fructose 1-phosphate and critical steps in converting dietary fructose into actual usable energy. Some of the associated diseases is HFI, or this hereditary fructose intolerance, and this is a genetic disorder caused by a mutation of that aldolase B gene. This leads to a deficiency in aldolase B. Individuals with HFI cannot properly metabolize fructose since it is in charge of breaking it down into usable energy, which leads to a toxic accumulation of that fructose 1-phosphate in the liver, the kidneys, and the small intestine. You can actually see this in the image below when we're talking about hyperglycemia. So individuals that suffer from HFI will have severe hypoglycemia, which has vomiting, abdominal pain, liver damage, all following the indigestion of fructose or sucrose. To manage this, the main treatment is a strict dietary avoidance to any kind of fructose or sucrose. Then when we're talking about Aldolase C, the clinical significance of this is because it's predominantly expressed in the brain and it's involved in glycolysis of the neurons. It is critical for maintaining the energy applied to the brain, which is highly dependent on glucose metabolism. Some of the associated disorders are a neurodegenerative disorder, although direct mutations of this Aldolase C are rare. Altered expression levels of Aldolase C have been observed in certain neurodegenerative diseases such as Alzheimer's and ALS. These conditions may disrupt energy metabolism in the brain. There's another one called cerebellar ataxia. This one is still in the works. Some studies suggest that the deregulation of Aldolase C may be a contributing factor to people who suffer from cerebellar ataxia, which is a condition characterized by a lack of muscle coordination due to the cerebral dysfunction. Dysregulation of aldolase isoforms, especially aldolase B, can lead to metabolic disorders like that HFI, or the hereditary fructose intolerance. In these cases, patients are unable to process certain sugars, leading to toxic buildup. There could be dysregulation of the A and C, again having contributing factors to issues with or disorders of muscle and neurological issues. It can lead to muscle weakness, and it is implied in several neurodegenerative diseases. Diagnostic markers are elevated serum aldolase levels, particularly aldolase A, and can be used as a diagnostic marker for muscle damage, specifically for people who suffer from myopathies and anemia. It can also be indicative of liver disorder in some cases for people with a dysfunction of their aldolase B. In summary, the clinical significance of aldolase isoforms lies in their essential roles in energy metabolism across different tissues. Dysregulation of these isoforms can lead to various diseases, as we've already underlined. Understanding the regulation and expression patterns of these isoforms is crucial for diagnosing and managing related metabolic, muscular, and neurogenic conditions. To summarize, we covered glycolysis, gluconeogenesis pathways, the roles of aldolase in those pathways, the different isoforms of aldolase, and how transcription factors regulate aldolase B. This understanding is essential for appreciating how our bodies manage energy production and glucose homeostasis. This information is also key to understanding what happens throughout the body when there is a dysregulation of those aldolase isoforms, and why it's vital to study the systems that they are associated with. Some of this information is difficult to understand and take in. I've attached a couple of links that may be beneficial to use as a supplement to increase and or solidify your understanding. Again, there's a lot of acronyms, there's a lot of words that had numbers associated with them, and to try to follow all of that is quite difficult. This by no means is a substitution, but sometimes videos and different images are beneficial as a supplement in the learning process. And here's a list of my various references used throughout this presentation. This concludes my presentation, and I'd be happy to answer any questions that you may have. Thank you.